So I have the privilege to give the first lecture. In my lecture, I will frame a bit the work that we have done in the context of the Be Good project. The title is Why Are Honeybees Dying? And Sustainable Perspectives Offered by Advanced Technologies. You see, the title is a bit different from what was in our program because I extended it with, let's say, Marcus's selection, which is, let's say, my own research topic at Ghent University, and also it was an important part of our discoveries under Be Good. So Be Good stands for Giving Beekeeping Guidance by Computational Assisted Decision Making. It is a Horizon 2020 project, 17 partners coming from 13 countries, multidisciplinary. And we start on the 1st of June 2019, and we are now at month 53 from in total 54. We have an extension of 6 more months to finish it. Overall aim is to assist beekeepers in keeping their colonies healthy by developing technologies that will make beekeeping more sustainable. I have made, let's say, a kind of scheme that explains it. So we are relying here on big data coming from sensors, all kind of data, and they will be processed, and then something comes out of the processing. Sometimes it means that beekeeper have to be alert about the colony's health going down, and he has to intervene. And then an alert was sent to the beekeeper so that he can take action. The computational in our title refers to the fact that computers will be used to do the processing. The guidance refers to the fact that we want to give guidance to beekeepers by sending out alerts to warn them about what's happening in the colony. I will in my talk, I will tell a bit about colony mortality, colony lossless, and how it is explained. I think that there is some improvement possible there, and we have some ideas, and I would like to share it with you. Here you see the data of the annual survey of Colosse about winter losses, and Belgium at that time had quite bad figures with almost 1/3 of our colonies that were dying over winter. This is a paragraph that I cut from a very recent paper where, as an introduction of that paper and explaining the let's say the colony losses, framing the colony losses. So it said this phenomenon has been linked to the exposure of bees to multiple stresses in their environment. Indeed, several abiotic and abiotic stressors interact with bees in a synergistic or antagonistic way. And so this is how we explain the colony losses. And I must admit, I do it as exactly the same. Also in my papers, you find this kind of statements. So we say colonies, they are exposed to stresses, and then the colony is weakened, and because of that, they don't survive over winter and they die. However, there are these papers of these surviving colonies, and we are all very well aware of that. And then the first one was of Ingemar Vries, of the Gotland bees, 150 colonies put on an island and left untreated, and about 5 colonies survive, and they are the genetic basis of repopulation of the whole island. Yves Leconte's story about Avignon, where there were no honeybees anymore in the wild. After a few years, after 10 years, they reappeared again, and he made a very nice paper on that. And then we have the description of the Norwegian honeybee colonies that survived after a process of natural selection. This is a bit more recently. So what is then, in fact, the consequences of the events? So you could say that most colonies, when they are exposed to stresses, that they are weakened and that they die eventually, but that there are also survivors when they are exposed to stresses, they remain healthy and they simply survive. So I believe the drawing shows that so there is something wrong. We are missing a bit the logic of it. And I think here in the first, is it really that simply most colonies and survivors? Is that the wording that we should use here? I don't think it is. I think we should say that bees that are under an intensive beekeeping program, and this for several generations, that is what is happening on the top. And then the lower line is for those that are exposed to natural selection. So this is more precise, I think, to explain what really happened. And then is it indeed still as such that stressors, they weaken them or they make them healthy, although the stressors are exactly the same. So this is also a bit weird. So what I did, I flipped this drawing and I changed the wording into vulnerable and resilient. The intensive beekeeping results in vulnerable bees. When they are exposed to stressors, they die. And natural selection results in resilient bees. And when they are exposed to exactly the same stressors, they survive. What I tell you is not something new. It is, I think many of us, they see it like this, but I wanted to just to put it in the right order. The mechanism behind it, how do these vulnerable colonies die and then eventually die? The mechanism, stress caused the mortality of the vulnerable bees, is that that the generation of winter bees is weakened by these stressors because they are vulnerable, and this has an impact on their longevity, but also the previous generation, and I call it the pre-ultimate generation, is of importance, because when they are weakened, they will reduce the brood care of the ultimate generation, so that eventually, let's say, the ultimate will die somewhere too early in the winter. And what is then really happening for those natural selection colonies? How do they become resilient? The mechanism is in fact that they are locally adapted. So the population, it will follow the dynamics of the environment with respect to the vegetation and climate. So in the north, for instance, in Belgium, it is very typical. Normally, our black bees, they don't develop after the first sunshine. But there was a selection program to have bees that develop early for the early pollination services. So there is at that moment, let's say, no more connection with what happens in the environment, because if they develop brood too early and then there is afterwards, let's say, a bad period, then they have problems to collect sufficient proteins for that brood. But there are also protective traits, individual, like suppressed mite reproduction, immunity, detoxification, or behavioral traits like hygiene and grooming behavior. So what then about the beekeeper's intervention? Well, what they do is in fact a kind of recovery, and I try to explain it like this. There is somewhere an equilibrium that should be reached. On the one hand, you have the stress level where the bees are exposed to. And on the other hand, you have all these things that are genetically determined, where we have a local adaptation and these protective responses that I just explained. And there is a gap between that, and there is no equilibrium reached. And what the beekeepers do is, in fact, to recover and to try to reach that equilibrium. However, we just saw that not all bees are in the same position. There is a scale of resilience. They can be fully resilient, or they can be fully vulnerable, and everything in between that. So the real graph should be a bit like this, where you have this gradient in local adaptation and protective traits, and on the far left end you have the most resilient ones, and on the right hand side you have the vulnerable honeybees. And the recovery, however, that we do for our by the beekeepers, they treat all colonies exactly the same. And it means that on the left-hand side, you have colonies that are over-treated, in fact. And on the right-hand side, you have some colonies that are not sufficiently recovered and that eventually will die. So what are our options now? Yeah, we can continue as before. However, we will continue to have winter losses. It will ask for a lot of workload. Productivity will be good, but resilience will be bad. Or we can choose for natural beekeeping. And then indeed, winter losses eventually will be solved. Workload is very low. Productivity, however, here that's a major problem. And these bees, they will become resilient. So what we do here in the colors is, in bee goods, sorry. It's, well, it's in colors maybe as well, of course. But our focus was on advanced technologies. And I think that we can give a green light on all these levels of questions. So, in fact, what we want to do with Be Good is that the intervention of the beekeeper should become tailor-made. And so it is that we flip from this graph to this graph. We see how the health status of the colony is changing and then determine how much intervention is needed. And we give them as much intervention as needed. So it means that on the complete right-hand side, it will be a lot of intervention. On the complete left-hand side, we will give only little or none intervention. And to reach that equilibrium, that's a bit where we are about. How will we do that? So we use here hive monitoring systems, and this is a scheme that gives an overall approach of the Be Good project. So on the left-hand side, you see all these arrows going to the cloud. These are the data streams that are created from sensors put in a colony. Sensors on top, you see the weight. Then to the left-hand side, you have temperature. You have sound, but there is also vibration, gases, the gases that are measured. You have the bee counter where you count the incoming and outgoing bees. And you have weather station. All this information is sent to a cloud. And then let's say we develop algorithms that use as a reference our digital bee colony model. Bee colony model that is put inside a dynamic landscape model. And based on that, we determine whether an alert should be sent or not. Is that colony in danger? Should the beekeeper intervene or not? So the alert is sent, and we give them guidance. And then based on the business model, let's say what they exactly will do, that can differ. It is not only what the data coming from the sensors put in a hive, but in our project, we have also laboratory analysis for bee diseases. We measure colony strength on the apiary. We have developed devices so that we can measure pesticide contamination on the apiary. And we have developed tech tools for genotyping in the lab. And so for the forthcoming, the next presentations, the lecture of Maarten Schoonman will be about these sensors and about sending alert and giving guidance. The lecture of Martin Bensik is more specific on the vibrational data that is all recorded. The lecture of Elżbieta Ziolkowska is about a dynamic landscape model. And the lecture of Xiaodong Duan is about the algorithm that we are developing. And then the final lecture shortly after noon from Wim Verbeek. He will talk about socioeconomics from the beekeeping business. Yes. So let's come back to the hive monitoring systems. How can that be implemented in beekeeping practice? How should these interpretation go? I think there are three levels. First of all, is it the interpretation of the data curve? This is mostly monoparametric. It's just following one parameter. It is something easily for the beekeeper, but it has limited predicted power. So you see here some, let's say, curves that go down when they use the stock in a colony. You have the daily patterns there below it on the left-hand side. You have the nectar flow when the nectar is coming in. But we have also rubbery, which is a bit different, the curve, than when we have swarming, but because then at once, let's say, a major part of the colony will fly away. And then when the colony is inactive. So this is a very simple way of interpretation. However, so these are some examples of the curve going up or the curve going down of the nectar use or flow. And this is, let's say, a paper that I found that already from 2015 that describes, let's say, how measurement of temperature, sound, video at the hive entrance and weight can give information about these different aspects of the colony listed on the left-hand side. And so you see under, for instance, temperature, y-y, referring to the fact that yes, in winter temperature can be used for that measurement on the right, and also in summer. So this is how the table is designed. I will not go over the details of the paper, but let's say there are very good reference papers that describe how the first level can be completed. The second level, I call it computational assistant guided. Here it's a bit more difficult. Here we will develop algorithms, sometimes just for one parameter, but it can be also a combination of some parameters. This is more something that we offer to the beekeepers as a service. And the interesting thing, it has a predictive value, a predictive power. It means that it is not that you found that your colonies is swarming when they are swarming, but you can predict the swarming in advance. And this is the paper of the group of Martin Bensick from 2020, when they came up with an algorithm where they can predict the swarming 30 days prior to the event, and this with an accuracy of 90%. So then it becomes interesting. You know it in advance. Our approaches at BeeGood, there are two approaches that we are following. One is where we are looking for outliers, and it seems that this gives a good prediction about colonies that are in trouble. Another one is a more integration of selected data sets. We have used the data coming from our colonies in Belgium and the UK, weight, in-hive temperature and out-hive temperature. And then a model was built to predict winter mortality, and we could predict it with an accuracy of 88.9%. So this was quite satisfying. Let's see. The third level is something novel, invented by the group of Martin Bensick. And I think we were in our consortium the first, or at least I think we were the first with this approach. It is elicited responses. It means that you trigger the colony and see how they respond. It is also something that you offer as a service. It has a highly predictive power. And in the example, and Martin Bensick, he will explain that very in detail, let's say, in his presentation. He uses an electromagnetic shaker. And so to deliver an oscillatory vibration to the colony, this is on the complete left-hand photo. You see that on the outside of the colony, that machine, the shaker, and then inside the colony, in the wax combs, you have this accelerometer. It measures the vibration in the matrix, and it will give a certain pattern. It will first show let's say, the peak that was created by the knock, and then something else. And what comes after the peak is what is important. You have first, let's say, a moment of reduced buzzing, and then you have a wave, you could call it. And so the interpretation of this curve helps you to tell something about the status of the colony. And then I would like to switch over to, let's say, the molecular marker assist selection as a technologies to give sustainable perspectives. Here I show you the life cycle of the varroa mite, but you all know that very well. Suppressed mite reproduction is a trait where the mite the mother mite is incapable of reproducing in the sealed brood cell, and this had quite an impact on the dynamics of the varroa population. So it is an important trait. So a study was done already quite some years ago, or started quite some years ago. We seeked for some colonies that were exposed to, that expressed already the varroa resistance in Europe. We have colonies from the Oostlandet region, in Norway, in Amsterdam water dunes, in Holland, in Capelle, in Belgian beekeeper, in Toulouse, were the bees from John Keefus, and then our reference were the bees that are sensitive in Ghent. We crossed them in twice, and then in the F2 generation, the drones, you have a segregation of the alleles. So, or they have the, let's say, the green resistant allele coming from grandmother, or they have the blue sensitive allele coming from grandfather. And then we start counting first the phenotype. We measured the percentage of non-reproducing mites. And it was found that the queens coming from the Amsterdam water dunes, they had the highest value. They have 50% non-reproducing mites. This was the highest figure that we could have, because it was a hybrid colony. You remember it was crossed in. And then we did some molecular work. So the pupae, they were Illumina sequenced for the whole exome. 64 drones, half of them were SMR positive, the other half were SMR negative. And we found 140,000 variants, and then some statistics was done. And at the end, we had a model of 8 variants that were associated with suppressed mite reproduction. Great. So this table explains the different SNPs. One point mutation that single nucleotide polymorphism or SNPs that were found. So 8 of them could explain the phenotype. Indeed, so this is a cross table on top. You see the model, what the model predicts. So you go and look at the genes that were found and the point mutation that were found, whether they were, let's say, the variant type or whether they were the wild type. And based on that, you predict what the phenotype was. You see that we found, let's say, altogether 30 that were affected. That's on the bottom line. These are the SMRs predicted. But then you see what is the reality, which of them were really SMR positive. And then you see only 27. And the same for the control. So in total, 56 of our 64 were correctly predicted by the model. So that's an accuracy of 88%. So that was quite promising. So in Belgium, the alleles are widespread. We found more risk mutations than protective ones, and that's normal because in Belgium, let's say, our bees suffer much from varroa mites, so they don't have, let's say, the good protective traits. And then what was done in the context of the Be Good project was that we developed here our TACMAN assay for rapid screening. So it's not the idea that everybody will do genome sequencing of all exome sequencing to find the mutations. So we have a laboratory test to do that. And it is based on a probe, a TACMAN probe. You see it on top of the sheen. It has a fluorophore and a quencher. And when it matches perfectly really well with your template, the DNA of your sample, then let's say the polymerase will break down the probe and cleave it so that you have a separation of your fluorophore and a quencher. When they are all on the probe, then the quencher prevents the fluorophore of sending out a fluorescent signal. When they are broken down, the signal can be sent out. So this is so you will see a buildup of fluorescent signal during the execution of this test. So when it works very well, you can find single point mutations that are or from the wild type or from the variant type. And so let's say just with 8 assays, you can screen your colony for the variants that are associated with the protective trait. Regis Lefebvre, he is one of my PhD students. He has done the reality check in Flanders just to see whether in the complex situation of Flanders with the different bee races, the different beekeeping techniques, and maybe also the different landscapes, whether this model that we developed still holds stand. He sampled 328 brood samples. He opened brood cells. He was screening for SMR positive and negative. Eventually, he had 842 drone pupae, and with 232 SMR positive and 610 SMR negative. And he phenotyped, so they were phenotyped, of course, and then they were also genotyped, and then he ran a singular variant test, and he found that the SNP number 2, 4, and 6, they were associated with the SMR. And then he did some logistic regression, and he found new variant models, a model with the 8 SNPs in, and a model with 3 SNPs, 3 variants in. And the accuracy of this 3 variant model was 87, so 730 of the 842 pupae could be predicted positively based on the genotyping data. So this was quite promising. It is normal that the data coming from one colony cannot transfer to the whole to a whole population. You have to recalculate your model, but you can use the SNPs that were found at first, and you don't have to do the whole genome sequencing for, let's say, your next level screening. What I here call the reality check. So in conclusion, we gave a bit a new concept of colony losses, although I believe that most of you are already convinced that this is a bit how things goes in reality. We believe that beekeepers' intervention should become tailor-made, not too much, not too little, just enough, and that hive monitoring systems can assist here. The implementation in beekeeping practice can occur at 3 levels. We showed that, and in BeGood, we demonstrated that the proof of concept for level 2 and 3, they really look very promising. We believe also that we should shift to a more resilient bees. Marker-assisted selection can assist here, and a 3 variant model of SMR trait looks very promising. So work on marker-assisted selection was mainly done, yes, it was exclusively done, in fact, in my lab. That's why I would like quickly to show the people that were involved. So I referred already to Regis Lefebvre doing the molecular work. Emma Bossuit present here today. She will work on the practical implementation in our selection programs in Flanders. David Kleijs-Boaar, he finished his PhD, and he was the one who was involved in the developing of the Takman essay. And then we had support from Ellen Daniels and Lina de Smet. We are also working together with the laboratory of Luc Pelman and Bart Brooks, and Mario Van Poeken. He assisted us there. This was a nice picture from the Be Good Consortium. I believe it was taken in Bologna in one of our consortium meetings. Thank you for your attention. And Emma, did I keep time, or did you forget to keep time? How much do we still have? Okay, we have all 2 minutes. 2 minutes left, I believe. Okay, fine. Are there any questions about my presentation? Yes, Alexis. Wait a second, we give you the microphone.
Hello. Yeah, thank you.
Yeah, just 2 quick questions. At the beginning of your talk, you mentioned that there is this differences between the intensive beekeeping, where we end up with colony losses that may be also partially caused by it, and also natural beekeeping, or no interference with natural selection. There's I think there may be also an in-between, or several in-betweens, where there are beekeepers who don't have so many intervention in their hives. So what do you see there? Like what could be, because these beekeepers also have quite a lot of colony losses in the end, although they may have a more an approach that's a bit closer to natural selection because they don't do so much interference. So what do you see there? What could we add that to the model, and how?
Well, but I think in the graph that I showed you later on with the equilibrium point and the arrow going from the stresses going from the top and then the arrows going from the bottom up. I think there I show that I don't see this black and white. I see it as a continuum coming from extremely resilient to extremely vulnerable. And our colonies of the beekeepers, they are somewhere in that gradient of resilience. Some have done investments in the sense of screening for specific traits, and they were shifted up to the left. But some of them, they are not that interested in resilience, and because of that, maybe they shift more to the right. So I think it is a matter of there is in our population, clearly there is in our population the genetic potential of returning back to the resilient state. But let's say we have to include our screening somewhere in our selection processes, so that it's become part of the selection, and that not only we focus on, let's say, what they did 50 years ago on honey production and on calmness of the bees. You know that with Carnicas in Belgium, and I think in many countries, you can really work on the bees just in a t-shirt without any protection. And this is, let's say, most probably because of the origin of the bees, but also of an investment for many years by the beekeepers to do that. And maybe that's okay. In Flanders, it's very okay because we are a very overcrowded country and too many people, and meaning that if you would have bees like they are in Spain, where you have to keep distance to approach a colony because they are quite aggressive, I mean, it would not work in Belgium. But now we have to invest in returning to the resilient state, and the genetics are there, but we have to increase the allele frequency of these protective alleles. I think that's important.
Yeah, that leads to my second question. I totally agree with the marker-assisted approach. But I remember from my previous postdoc in Avignon, there was this big project running at the same time called Be Strong, where they were screening lots of colonies in France and looking for the SMR trait. And they did that over several years as well. And they ended up with a very big data set where they could compare as well within the same colonies the value of this phenotype. and they tested also the repetition and the repeatability of the test for the phenotypic assessment. And I just checked now, they found a repeatability of 0.4 only of SMR in the colonies, which is not super high. So I was wondering, how do you consider a colony to be SMR? At which level of mite non-reproduction do you say this one is SMR, this one is not?
Well, the screening is dus the model can predict, let's say, of individual pupae the status, but it can also give the allele frequency of the different variants in a colony or of the queen. So this is how we work. If we want to know the genetics of the mother mite or of the queen in a colony, we use the legs of drones, and we put together in a pool, let's say 30 legs of different drone pupae, and then measure whether they are homozygous. The queen is homozygous for the variant or the wild type, or it's heterologous. So this for the HSNP. So you can let's say determine the genotype of the mother quite precisely without using the queen itself. I mean, you don't have to sample her. You could do that also with the wing cut, but we prefer not to do that. And I think that what we are now doing with the drone pupae legs is very gentle. If you take worker bees, then you have, let's say, more the status of the whole colony. Now, so the word where you refer to was about phenotyping, I think. It was not about genotyping. So I think we have to understand that marker-resistant election and the model, the ideal model that we predict or that we determine, that it is not something where you can rely on for 100% for individual colonies. It is more, let's say, that we see in the whole population that the model there still works. The genotyping can do a very good prediction of SMR. And that in some cases where a beekeeper do the genotyping, and based on that, let's say, he will do a certain crossing to improve the status, the genetics. It will be followed by an improvement also of the expression of the phenotype, but in some cases it won't. It is more at the population overarching level that the let's say the accumulation, the increase of the density of the protective alleles will eventually also be accompanied by an improvement overall. I think that's how we would look. Secondly, I think we are at the start of the marker-assist selection development, and there is a lot of things we still don't know. And I have demonstrated here something for one set of variants that work, but might very well be that when I do exactly the same in the south of France, that I find a completely different set, and that we in let's say 20 years we have a few hundred, a few thousand of interesting variants, but that we then, let's say, most probably don't do individual testing anymore by Tachman assay, but by then maybe the whole genome sequencing cost €1 or something like that, and that we are then have access to this approach and take into account all these different variants that we think that contribute to resilience of the bees. So we are just at the start of it. Yeah.
There's an online question, Dirk, and I think James, do you want to step in on that one?
Yes, sorry, I was just trying to find my unmute button. Yes, we have a question from Miha Finsgaard. I hope I pronounced your name correct, but dear Professor Dr. de Graaf, do we know what the effects of different hive monitoring systems on the bee colonies are? How electromagnetic and other signals interfere with the colony's well-being or even their resilience? Thank you.
This is a good question. Mostly that's my students always also say when I ask them a question during the exam and they don't know the answer, then they say that's a good question. So now I also say this is a good question because this is the first time that somebody confronted me with this. But Martin Schoonman, do you have data about that?
Yeah, so it's a very valid question because we use hive monitoring and data is transmitted wirelessly, and we want the colonies to be healthy. We also don't want this transmission to interfere with the experiments. We have not researched this specifically in Beekeeping. I know others have researched this, and Martin Bensik is pointing his finger regarding this. A short one, yeah.
Yeah, briefly, there is one study I know of with someone putting a mobile phone on the top of a hive, and they claimed they had some responses. That's the one study I know of. And in my opinion, it's a very, very limited study with only one hive, one instance. And the other thing I can say is that I have kept honeybee hives on my university campus with thousands of students holding their own mobile phone on them. The university campus is absolutely polluted with electromagnetic waves coming from wireless human-made transmission systems. And my bees on campus do just as well, if not better, than in remote areas where there are no electromagnetic waves whatsoever. So data suggests so far that those electromagnetic waves that we use for wireless communication purposes have no measurable effects on honeybees, unless perhaps they're in the immediate vicinity of a high power antenna responsible for centralizing those waves. But provided you're far away from one of these masts, in my opinion and in my experience, you should be absolutely fine.
There is a question here in the...
Yeah, I think this will be the last one because of time. And then we will move on.
Norman Carrick, it was just to add to what Martin said there. Yes, there are a couple of published studies on this which were very badly done, and I don't think you can draw any conclusions. But I do know that Thierry Blaquier at Wageningen did do an experiment where he put some hives in a Faraday cage and other hives that were outside and exposed to electromagnetic radiation. And, you know, as far as he was concerned, there was no significant difference. So, you know, clearly that's a negative result. So you can't draw permanent conclusions. But I would agree with Martin that so far there doesn't appear to be any evidence that there are effects.
Thank you. Counting.
Hello, I'm Fanny. Well, there are 4 Horizon projects now. One is called E10, and there is about electromagnetic radiation on human health. But this one, particular at E10, it has 2 work packages working on insects and honeybees. So we are going really to do, we start now, just yesterday, I installed the antennas on the laboratory, then we will do the field. So we're hoping that we can see things. We don't know, but we agree that the studies before were not according to what they should have done for this. So we will see. And now we go to 5G, which is 5 times more.
Yeah, okay. Thank you. Shall we move? Yeah.
Sorry. I was thinking when I was listening to your presentation and you're talking about stressors and resilience, what I hear if I listen with my beekeeper ears is stressors synonymous with varroa mite and resilience to varroa mite. If we leave out lack of local adaptation, how much would you say, like what percentage of stressors do you think is varroa and what is everything else? Just one number to make it very short.
Well, but I think if we really want to go to a situation where your number of interventions is really absolutely the minimum, I think then the local adaptation plays a very big role. If you allow, let's say, the constant follow-up of a colony to see whether they have food and so on, and to feed them supplements if they are in shortage and so on. Then, of course, then the local adaptation is something that doesn't need to be that much present because the beekeeper does that intervention. But in my gradient curve, then they are a bit, yeah, let's say somewhere in the middle because they are lacking too much local adaptation. I think if you want absolutely the match between the dynamics of the colony and the dynamics of how the environment is, then you're reaching that point of absolute local adaptation and they are more to the left. And if on top of that, they are, is that satisfy your question or not? I see yours. This is the opposite. Okay. Well, I think at this moment, Varroa is the key player of the losses that we have for sure. And so that's why I believe that in the introduction of traits to cope with Varroa, but also traits to cope with the viruses, they are utmost important at this moment. Yes, for sure. Yeah.